Great. Okay, I think we can get started. Hello, everyone, and welcome to this webinar entitled Novel Engineered Polymerases for the Detection of Infectious Disease. I think this is going to be a really interesting webinar, so it's great that you can be here today. Now, before we get going, just want to say that this webinar does have a Q&A function. So as we're running through the webinar, if you have any questions, please just click the Q&A button at the bottom of your screen, submit your question, and we'll try our best to answer your question at the end of the webinar. As well as this, the webinar is being recorded, and the recording will be sent out to everyone who registered for this webinar. So please feel free to watch this again if you find it interesting, or pass it on to your colleague if you think they'd find it valuable. Now, before I introduce our speaker for today, I just want to quickly introduce who we are. So MedixMDx manufactures raw materials for IVD manufacturers. And the company started in Finland over 35 years ago, and since then has continued to grow and acquire new companies. And we now have offices in the US, in Finland, Germany, France, and China. And we've now grown to a size where we're providing raw materials to almost all IVD manufacturers around the world. Now, you'll see that I've highlighted in red here Constance, Germany. That's because this is where the MedixMDx business is based. And MedixMDx is Medix Biochemica's molecular diagnostics business. And we specialize in engineering polymerases for a wide range of DNA and RNA applications. So basically, engineering polymerases to have interesting characteristics to solve problems. Now, as well as the off-the-shelf products that we provide, we also provide services like assay development services and lyophilization services. But it really is these engineered polymerases that we want to talk about today and their role and impact in the infectious disease space. So with that being said, I would like to introduce our speaker for today, Dr. Maja Studencka-Turski, who is our scientific lead of the R&D team in Constance and really is one of our experts on the MedixMDx products. So welcome, Maja, and please take it away.
Hello, everyone. Let me just share my screen with you. Just a second. Can you see my screen?
Yes, I can. Just need to put it into full screen mode.
Yeah. Perfect.
Yeah.
So hello, everyone. Thank you very much for the introduction. So my aim today is to tell you something about why we actually engineer polymerases and how is this relevant for diagnostic of infectious diseases. Then I will go to explaining you how we generate polymerases, and I would like to introduce you to products in our portfolio, products which were engineered by us and developed with, and I will show you some examples of applications how you can apply these products to diagnostic of infectious diseases. So, as most of you know, you cannot imagine modern molecular biology and biotechnology without polymerases. So they are absolutely essential for processes such as DNA cloning, simple PCR reaction, but also DNA sequencing, more sophisticated techniques like synthetic biology, molecular diagnostics, and, for example, SNP detection. So for all of these methods, we absolutely need functional polymerases. And as you know, polymerases are these natural molecular machines, so we can isolate them from thermophilic bacteria. And they usually, except of that they are very thermostable, they have a broad range of very unique characteristics. So they are very unique in terms of the set of the characteristics they are having. For example, they may have a different extension rate, so which means the speed at which they are adding nucleotide during DNA synthesis. They differ also in terms of the fidelity. processivity can be different, how long they bind to the DNA strand. Some of them are more specific, some of them are less specific. They are able sometimes also bypass the DNA lesions. If they have an additional domain, the nuclease domain, then they are able also to, they have a nuclease activity. And some of them I also have this very nice feature of strand displacement activity. So the problem with natural DNA polymerases or polymerases in general is that their performance qualities are not at this standard that we need them to be for molecular diagnostics. And this is where we as a company step in and we engineer polymerases. So what we actually do, we can address any of these characteristics and improve the engineered polymerase so that it can be useful for broad range of molecular diagnostic applications, but also can be developed towards a very specific assay for molecular diagnostic. So how do we do that? Usually we are using two strategies. The first one is developing specific DNA polymerases or polymerases in general via random mutagenesis. So this is a method where we are taking a wild-type polymerase, we are introducing mutations via error-prone PCR. So these mutations are introduced in a totally random manner, and we come up with the thousands of mutants. We are building a library of these mutants, of thousands of such mutants, and then we are proceeding with the screening of these mutants with a particular assay of interest. Alternative approach is so-called rational design approach. This absolutely requires that the enzyme, that we have enough knowledge about the enzyme. Let's say we know, for example, which amino acids are responsible for increased thermostability or increased sensitivity of the enzyme or processivity. So by knowing this, we can introduce point mutation via, for example, site-directed mutagenesis. And by this, we can engineer an enzyme and develop it into specific application. A good example of engineered enzyme is a Clentac polymerase. So this is truncated version of Tac polymerase. It has increased sensitivity to inhibitors from blood, for example. It also has increased fidelity. So using these tools, we are developing enzymes which can be also applied in a diagnostic of infectious disease. But what actually does that mean? What does it mean that the enzyme is good for application infection disease? So there are certain features that we always have to focus on developing such an enzyme or engineering it. What we are seeing, it's important that we are having enzyme which is sensitive and specific. So sensitivity is crucial for such an enzyme when we are applying the enzyme or using in the molecular diagnostic of infectious diseases, because if the enzyme is not sensitive enough, then we are dealing with, we are not reaching the target levels, so we are not detecting the, for example, the target of interest, which means that we may obtain false negative results. If the enzyme is not specific enough, then we are running into the likelihood of receiving false positive results. The other important feature is that the enzyme should be fast enough. So it should have a fast, high processivity, because this is obviously crucial in a diagnostic of infectious disease, that the enzyme, that we can get the results as fast as possible. Moreover, if the enzyme is specific and if it's inhibitor resistant, then we can completely skip the, we can work, for example, directly with crude samples. So we do not need to extract DNA or RNA. We can work with direct application, let's say with the blood or with plasma, and we can try to detect infection in the samples directly. So this is also another feature which saves a lot of time during the diagnostic. Another important point is that such an enzyme should be able to detect broad range of pathogens. It should ideally work in a multiplex system, so it should be able to detect many targets at the same time. Moreover, important for molecular diagnostic is the stability of the enzyme. So this enzyme ideally should be stable in room temperature, so it makes diagnostic tests easier to distribute, but also it's able, we can use such an enzyme, such engineered polymerase, in, for example, in the field diagnostics where there's no laboratory available. And if we have such an enzyme, it's also ideal for application in point-of-care testing. And last but not least, the engineered enzymes should be also able to be applied in advanced technologies like NGS, for example, or CRISPR technology. So let me introduce you to 3 different types of products that we are having in our portfolio. All of them contain engineered polymerases. This is a PlexTag 5x qPCR multiplex master mix. I will show you 2 products for RNA applications, and lastly, I will introduce you to 2 products in our portfolio which are ideal for isothermal amplifications. So let's start first with PlexTag mix. PlexTag is our all-in-one, very unique engineered product. It contains formulated polymerase, tag polymerase. The mix is 5 times concentrated, which means it gives you a lot of free volume for the sample. And it really matters because this product is also compatible with crude samples, which means you don't have to worry that you are not having enough of sample in your test vial. So if you have a very concentrated mix, then you have a lot of space for the test sample. It's also highly specific, and lastly, it's lyo ready. It's ready to use for lyophilization. You don't have to add any additional excipients. So let me show you an example how PlexTag performs directly with blood. So in a traditional approach, one would obtain a blood sample, and then we would proceed with DNA extraction from such a sample. And lastly, we would use the isolated DNA in a qPCR application or assay. When you're using PlexTag, you can completely skip the DNA extraction step. You can use blood sample in your reaction and proceed with qPCR directly. So there are no reagents required for DNA extraction, also no equipment required. It saves you a lot of time and is less error prone. To show you an example how PlexTag performs with blood. This is an example of a multiplex with 3 targets. We detected these 3 targets in a dilution series of samples starting from 0.1% of blood up to 20% of blood. So you can see detection of these 3 targets depending on the amount of blood in the sample. So I would like to draw your attention to the bottom left, where there is an internal control target. So this is a target. We spike the blood with 10,000 copies of synthetic DNA to have a target which is the same, with the same amount in every sample. So what you can see in this graph, that basically the detection is not changed. What is changed is the intensity of the signal. So the polymerase is still able to amplify the target even if there is a 20% of blood in the sample. It also amplifies very nicely targets coming from the blood, so coagulation factor 5 and actin as a housekeeper. So you can see very nice amplification. Of course, it's strongly dependent. Signal intensity is dependent on the amount of blood in the sample. But to prove you that there is amplification taking place, we loaded the samples on agarose gel, and on the left you can see comparison between 0.5% of blood in the sample, and you can see the detection of these 3 targets of interest. And next to it, you have the same targets being amplified in 20% of blood in a sample. So you can see that basically the amplification is equally good regardless if there is a 0.5% of blood or 20%. So PlexTag works very nicely with the blood samples. Another good example how one can use PlexTag, applying a PlexTag in a multiplexing, is an example coming from our collaborators. And this is a Cytec National Research Council in Italy. They did a comparative studies between PlexTag and some alternative mix, and they apply PlexTag in multiplex microarray. So in the first step, they used PlexTag for amplification of the 3 targets that they use in their multiplex microarray, and the alternative mix. They already noticed the difference at the very beginning because the PCR with PlexTag was 20 minutes shorter than for the alternative mix. Then they label the amplified DNA read reporters. They loaded the samples on the microarray plate, and they measured the fluorescence intensity. And this is a diagram showing you the fluorescence intensities for these 3 targets and the comparison between the alternative mix and the PlexTag. So you can see that for 2 of these 3 targets, the performance or the intensity of the signal is basically the same, whereas for 1 of the targets, for beta-actin, PlexTag outperformed the alternative mix. So it also shows you that not only that PlexTag can be not only used for qPCR, it also works very nicely in the microarray PCR approaches. So just to conclude on PlexTag, this is a really all-in-one mix, which is very easy to use. It's widely used workhorse mix for us. It can detect up to 30 targets in multiplexing. It is live ready. Very importantly, it is produced according to the ISO 13485 certification. It can be used with crude samples. It works very nicely with blood, with saliva, with plasma. Moreover, it can be also used for food pathogen testing in a lyophilized form. The second application and example I would like to show you are our products for RNA applications. And for RNA applications, we have a very interesting product. It's called Volcano RT-PCR Master Mix. Volcano stands for very hot reverse transcription, because basically what we did, we engineered Taq polymerase so that the Taq polymerase also has a reverse transcription function properties. It's able to simultaneously perform reverse transcription and amplifications. This is a ready-to-use mix. It's very thermostable. It contains Taq polymerase, which is hot formulated, so it's hot started. And it provides also reliable results with crude samples. So just to show you how hot is this hot reverse transcription, this is a comparison. This is a primer extension experiment where we compared a performance in a gradient of temperature between the standard reverse transcriptase and the Volcano reverse transcriptase. As you can see, the standard competitor usually does not perform reverse transcription in temperatures above 50 degrees, whereas Volcano, the optimal range for Volcano reverse transcription is between 60 and 70. What is the benefit of that? First of all, it gives much higher specificity, because at these temperatures, you are not dealing anymore with secondary structures of RNA. So it's very useful when you're using when you need something for application in virus screenings. Secondly, because reverse transcription takes place simultaneously with amplification, one can completely skip the isothermal RT step in qPCR. You can use so-called zero-step RT approach, where you can perform reverse transcription in the same time as the annealing. So it saves you also time for your assay. And to give you an example how Volcano can be used, these are examples coming from University of Miami. So University of Miami set up an approach based on our Volcano product to detect viruses, in particular SARS-CoV, HIV, and also monkeypox in wastewater screenings. So they were able to extract DNA from wastewater, and they detected they used this DNA, then RNA, sorry, for testing using Volcano. Moreover, they were also able to very nicely detect all quantitatively and qualitatively viruses like HIV and Zika virus. A second product in portfolio for detection of RNA is our MedixMDx qRT-PCR Lyo ready mix. So this mix was specifically designed for lyophilization, but this was also optimized for rapid detection and quantification of virus RNA, but also for total RNA and mRNA. It's very effective in multiplexing. So we always recommend it if you wish to detect more targets of the interest. So the difference between Volcano and the RT and this RT mix is that Volcano is a one enzyme having two features. So it can perform DNA amplification, but also reverse transcriptase. In this setup, we have a combination of two enzymes where there is a Taq polymerase, which is hot started, and a reverse transcriptase. And this reverse transcriptase performs reverse transcription in this up to 50 degrees temperature ranges. So to show you how what's the performance and the application example for this mix, these are diagrams showing you amplifications in 3 different serum sample quantities in comparison to other supplier for hepatitis virus A, B, C, and E. Please pay attention that virus hepatitis A, C, and E are RNA viruses, and B is DNA virus. So it's able to amplify, of course, also works very nicely with the DNA viruses. So you can see that in comparison to supplier A, we have a more reliable detection, specifically also in this lower amounts of serum samples. Just to conclude again, for this RNA amplifications. So we have 2 products in our portfolio. The Volcano product, so this is this product where we engineer polymerase so it can detect DNA and RNA. There is a reverse transcription taking place, but at higher temperature than normal transcription. This is a product which is also produced according to the ISO standards. It contains polymerase which is hot started, and importantly, this is also a product which is formulated so that it can be directly lyophilized. So it can be also lyophilized if the client wishes to have it in this form, or you can also lyophilize it yourself. The other mix contains 2 enzymes. So it contains Taq polymerase and reverse transcriptase. It's already also developed to be lyophilized, is ideal for multiplexing for viral detections. And lastly, I would like to introduce you to our products for isothermal amplifications. In this portfolio, we have very nice Medix Fast BST product line. This is perfectly suited for isothermal amplification. It can be applied for LAMP, but also for RT LAMP applications. It's very fast. I will show you the results in a second. It can be provided as a master mix, but also as a separate component. The second product in this portfolio is our isotherm 3G product line. This is a very unique polymerase because it's also engineered so it can use RNA as a template, not only DNA. So it can be applied in LAMP and RT LAMP amplifications and is ideal for point of care testing. So just to convince you why LAMP is worth of attention. So LAMP stands for loop-mediated isothermal amplifications. This is a low-cost alternative for detection of infectious diseases because it does not require any specific equipment. So the reaction is taking place in one temperature. There's no cycling, there is no denaturation of DNA. So of course you don't need any cycler for LAMP testing. It's also very fast. Usually you get results within 15 minutes. It's easy to visualize. So this is an example of LAMP for COVID samples. You can see the changes in the temperature, in the color from violet to blue for samples which are positive for COVID. So it's also very simple to operate, and because of that, it's ideal for point of care testing, but also for at-home use. So let me show you an example how fast is the BST, the fast BST family. I would like to draw your attention to the upper left corner to this diagram. It shows you the detection times for our Medix fast BST and the competitor. So for 4 different DNA concentrations, you can see that for every of these concentrations, BST is approximately 10 minutes faster than the competitor, but this fast time to results occurs without sacrificing the performance. As you can see on the right, we can detect even the lowest concentrations of DNA with this isotherm. Moreover, this fast BST product is also very resistant to inhibitors. So it's not really bothered if we add 0.5% of blood into the sample. Still, it performs the reaction approximately as fast as as without the blood. So it shows you again that it is in fact resistant to inhibitors, and it's very fast. It's faster than competitors. We use also the fast BST on, actually it was used by the Italian Institute of Technology. They use our fast BST for benchmarking. They wanted to have an alternative mix to detect Salmonella. They also specifically asked if we could provide them with warm start BST. So we developed warm start BST for them with UDG. And they use these 2 mixes, so the fast BST and the warm start BST in their testing, comparative testing with the competitor. So you can see that using 1,010 copies of Salmonella doesn't matter for fast BST, because it takes approximately 5 minutes to obtain results with this mix. For warm start BST, it's also very reliable. It detects 10 copies and 1,000 copies of Salmonella, and it's performing the reaction much faster and more reliable than the competitor warm start with UDG. The second product, which I mentioned, this is our engineered isotherm polymerase, which has improved reverse transcription activity. So there's no addition of reverse transcriptase needed. When you are using this isotherm, it's very nicely amplifying RNA targets. This is an example of performance of isotherm 3G using RNA for engine, for SARS-CoV engine, in comparison to the wild type BST. So you can see it needs only 50 minutes to apply the target. And on the right side, you see a benchmarking comparison with 2 competitors, where isotherm is the fastest from the competitors. It's almost is a little bit faster than the competitor 1. Also and is also as sensitive as competitor 1. However, it's not having the issue of false positive control samples. And when you have a look on the competitor 2, the competitor 2 it needs twice as much time for amplification of this RNA target. Isotherm can also be used for detection of DNA. So again, with the help of Italian Institute of Technology, we could perform this very nice comparison. Actually, they performed for us for Salmonella and Legionella targets, but they also tried some other bacteria, and the results were the same, where you can see application of isotherm for amplification of 10, 100, and 1,000 copies of the bacteria. In case of Salmonella, the performance was faster with the isotherm 3G. It was also faster for Legionella targets, regardless of if this was 10, 100, or 1,000 copies. So let me sum up for isothermal amplification. For this applications, we have two products in our portfolio. This is our Medix Fast BST and isotherm 3G. So isotherm 3G is engineered to perform LAMP and RT-LAMP. It can perform reverse transcription. Fast BST is also able to be applied for LAMP. It's ideal for point-of-care testing, and they are developed and produced according to ISO standards. They both are available as a mixes, but also as separate components. So at this point, I would like to thank you for your attention, and we will be happy to answer your questions. Rob, could you please take over?
Yeah, great. Thank you very much, Maja. That was great. So we're now going to move into the Q&A session. So for this, I would like to introduce Dr. Ariel Bryan, who is our scientific business specialist in the US, to help with the questions. So welcome, Ariel.
Hello, thank you, Rob.
Great. So let's move on to questions. Question here for Maja. When you say 20% blood in the sample, do you mean from PCR reaction volume, or how much blood percentage you add to your PCR mix?
I mean the PCR volume, PCR reaction volume, yes.
Great. Another question here for Maja. Are your polymerases fast? What is the minimum annealing extension time? What is total time for 40 PCR cycles?
The question is for which product is this question specifically? So for example, just give you an example for PlexStack. For example, if you're using PlexStack for pathogen detection for foodborne pathogen, it's enough. The reaction runs usually in it takes 15 minutes for one PCR, and this is for 50 cycles. Because for PlexStack, you don't need this initial denaturation time. You can directly start with cycling. You don't need this 2 minutes or 5 minutes, 95 degrees or 94, if you will. You can directly start with 10 seconds denaturation and then proceed with annealing time. So this saves you a lot of time already.
Great. Okay. Question for Ariel here. Do you have any experience with freeze drying isotherm 3G? I'm interested in carrying out LAMP on a test cartridge and would need isotherm 3G in enveopalized form.
So yes, actually, we're working on a master mix that is ready to use, Lyo ready, off the shelf. But we also have the enzyme available in a format that can be concentrated, custom for you. But please reach out if you have a specific application, and I can help facilitate a custom enzyme. But soon we will be releasing a master mix, which is both Lyo ready and warm start.
Yep.
Great. Another question here for Maja. Could you explain a little bit more about the engineering part of the presentation? I'm interested to know how you choose whether to go for random use genesis or rational design approach.
Yeah, that's a good question. This is actually, it really depends on what is our application. As Rob mentioned at the very beginning, we are also provide services or assay developments for our clients. And if they are, if you're looking for engineered polymerases for a particular application, then usually we look what we have in our stock available. If we have something which is already developed for such an application, and then we can sort of improve it using, for example, rational design. Or if we don't have an enzyme ideal for this application, then we would preferentially start with a random mutagenesis, and then we would have a look what came out of the screening. If the enzyme is, if we are satisfied with the, or client is satisfied with the performance of the enzyme, then we leave the enzyme as it is. But if we feel the enzyme still needs some improvement, let's say it's not fast enough, or it requires a better specificity, then we would, in addition, apply the rational design approach. We would introduce some point mutation if we have enough information about the enzyme, so that we can improve the performance of the enzyme.
Great. Okay. Another question for Ariel here. I'm based in the US. Do you sell directly in the US, or do you work through distributors?
Yes, we do sell directly in the US. So from that map at the beginning of the presentation, we have sites all over the world, and this includes a site in St. Louis. So if you're ordering an off-the-shelf product, we can ship directly to you. But right now we're in talks with several local distributors, and we hope to be able to announce those soon, which might be able to better serve if you're working with a purchasing platform or you're restricted on which distribution networks that you need to use. So get in touch, and I can let you know what's available to you.
Great. Okay. And probably have time for one more question. So got one more here for Maja. I'm interested in testing for RNA pathogens. You mentioned 2 products in this speech. Which one would you suggest?
It really depends what is your testing. If you're, I mean, generally, we would recommend to try with Volcano for viruses, because then the reverse transcription takes place in a higher temperature, so it will be more specific. But if you have, for example, an assay where you would like to detect more viruses, like 4 or 5 targets, then we would recommend to use the Liomix for that.
Brilliant. Okay, well, we've come up to time. So firstly, thank you to Maja for the speech earlier. That was great. And thank you, Ariel, for helping out with the questions at the end. Thank you to everyone who attended. I hope you found that valuable. Hope to see you again soon. Have a nice day. Goodbye.
Thank you. Bye.
Bye. Thank you.